I will just a brief introduction for myself. For those who don't know me, I'm Martin Middleditch. I'm a lead technologist at the University of Auckland's Mass Spectrometry facility in the Faculty of Science. So I run a facility that operates GCMS-based analyses for metabolomics and volatile-omics, as well as LCMS-based platform, which we use mainly for proteomics, but we get involved in other things too. So we have a membership of, you'll see in one of my slides, if I can, is my screen sharing still?
Yes,
It is.
sharing the screen, Martin. Thank you.
Okay. Um, and so let me just, yeah. So we have a facility that's part of the university's life sciences mass spectrometry platform. And so we have groups from Liggins Institute, Auckland Cancer Society Research, and medical school, all combining to sort of provide a platform for mass spectrometry analysis in the life sciences. But we're also part of the Mass Spectrometry Hub, a much wider grouping of just general mass spectrometry practitioners. And so, yeah, we serve a very wide community. Our facility alone, we're providing proteomic analyses for synthetic chemists, biologists, anthropology researchers, forensic scientists, biotech companies, food scientists, wastewater engineers, all kinds of stuff, and including hospitals, obviously, which is the reason for this talk today. So, yeah, just to focus on one of the things we do, which is this mass spectrometry based typing of amyloid biopsies. We'll just have a brief, let me just, here we go. A brief, possibly redundant slide for this audience, but just to have some key points stressed. So obviously, we're looking to uncover the causative proteins that are misfolding in these tissues. The diagnosis of the disease in general is often quite straightforward, but of course, the subtyping is the key part. There's obviously 30 or more proteins possibly responsible. And of course, the treatment depends on getting that correct identification. And a bit of history. Yeah, we've had about 14 years now. We've been involved in providing mass spectrometry-based typing. We've had about 263 cases to date from all kinds of tissue types that we've been working with. And to give a sense of that graphically, here's the sort of summary in terms of a tissue analysis. Over half of the tissues are from heart and kidney, as you can see on that right-hand side, but a lot of other different kinds of tissues. Abdominal fat pad obviously is quite attractive if you're trying to avoid performing a cardiac biopsy on a desperately unwell person, as well as bone marrow. And then that grey wedge, you can see there's 22 further tissues that we've had, obscure tissues for analysis. So it's been a range of things we've encountered over the years. And how do we approach it? Reductionist science is probably the best answer. It's a great example. So we have our patient amyloid deposit, a solid tissue. We'll be harvesting a subsample from that presented material. If it's FFPE blocks, we have to dewax with heptane. If it's a fresh abdominal fat pad, we'll instead try and wash away residual blood, which can be a problem for us. And then we try to use denaturing buffers and probe sonication to solubilize everything. So we've gone from solid now to liquid. And now that lets us use the enzyme trypsin to chop up the proteins into peptides. We typically employ a temperature-controlled microwave for this, and we usually apply an hour, but it can be overnight. We then clean up those peptides and concentrate them in a vacuum concentrator. And now it's time to take that complex mixture of peptides and separate them to some degree with C18 reverse phase chromatography. We then transfer from liquid phase to the gas phase using the electrospray ion source to get those peptides charged and into the mass spec. And then another step of reductionism, we then smash up those peptides into their fragments. And we've reached a point now where we can turn the corner and start to rebuild the picture by taking all those peptide fragments and building them back into peptide matches that we then collate into a list of protein hits from the tissue. And the key thing here for us now is to look within that catalog for things that are unusually highly abundant and concomitantly having aberrant cleavages, not from the protease which we've added trypsin. So we're looking for things that have been happening in vivo with regard to those cleavages. And that, of course, is our main way of determining what the actual monomer is that's causing the deposit. So it's a very reductionist method, but it seems to work. To give a picture of the actual harvesting, you can use unstained slides. It's not our preferred format, and it does tend to give us probably the most trouble in terms of amount of material. Things work better when the clinicians can give us a marked up slide that shows us a sub-region that we should be focusing on so we don't harvest healthy tissue. There's no point in shoveling healthy material into the machine. But the best ideal situation is having that FFPE block and being guided by the marked up slide to know where to subsample and get enough material to move forward. And again, yeah, if we do receive an abdominal fat pad in the fresh state, we can actually wash away some of the blood. If it's already been put into the FFPE block, we don't have that opportunity to avoid the negative impacts of albumin and hemoglobin contaminating the picture. A quick look at the front end of the system we're using. It's a SciEx Zenitov 7600 QTOF. So we've got down here our 3 drawers with the chilled autosampler trays. We have our autosampler needle on this movable arm that can go and open the drawers for us and grab samples out of the vials and inject them into this trapping valve setup. And then this box has got the micro LC pumps that pump at 6 microliters a minute, get our solvent gradients to bump the peptides off the trap and through this tube onto our C18 column, which is housed inside this column heater at 50 degrees. And the base of this column is actually screwed directly into the end of the electrode inside this ionization source on the front of the mass spec. And from there, our peptides are ionized and can get pulled into the vacuum system of the QTOF mass spec, which is off to the right here. A quick word just about the separation of peptides. We're using these superficially porous media to try and get nice sharp peaks. We're hoping to sort of resolve as many peptides as we can in these rather complex mixtures. There's thousands of peptides, and so we're trying to use a good system to get rapid resolution of these thousands of peptides. But of course, we're not using a UV detector, we're using a mass spec, so we don't actually need to resolve them all from each other. As long as we just spread those peptide signals across the time domain, we can get the mass spec a fair chance to identify as many peptides as possible. I'm not sure if anyone's already au fait with ESI, but I'll just describe quickly the process of converting from that liquid phase into the gas phase. So our C18 column screws onto the end of this electrode. We have a gas to nebulize that into an aerosol. 2 more gas heaters that dry off that solvent. And so these green enlarged images of a droplet show that there's a lot of charged particles inside. We've got 5,500 volts applied in this ion source. And so all our peptides we hope are charged. And what we're hoping to do is dry off that solvent, these little yellow balls. We want to get rid of them and condense down those aerosol droplets till they get smaller and smaller. The charge density on the surface becomes too much, and there's these little explosions that create smaller and smaller droplets. And the aim of the game is to just drive off all that solvent until you have just ideally these bare blue peptide ions that are being drawn into the mass spec by a magnetic field and are free to be then analyzed in the mass spectrometer. So this is the wider view now. We've got our autosampler tray open where the vials are. Here's our ion source with the column, and now this is the actual mass spec part where we've got our ion path and the flight tube inside this housing. So yeah, that's the actual setup from further away. Going inside the machine and trying to explain the concept of MSMS, if those who aren't that familiar with it need a little primer. We've got our charged peptides that we've formed in our ion source that need to go into the machine now and be focused through these lenses to the first quadrupole Q1. An initial phase, we want a survey scan to just see what the peptide masses are that are eluting from the column at that moment in time. So we gently pass the peptides through the collision cell without getting fragmented and allow them to be accelerated into this mirror gated ion path. And so we can actually measure the intact peptides molecular weights with a high degree of accuracy. So the machine will record the time taken to transit this time of flight chamber, and that will correspond to their mass to charge ratio. At the same time, that spectrum will show us what the charge state for each of those peptide species is, determined by its neighboring naturally occurring carbon-13 isotopes. So we get that piece of information as well. And now we tell the software that we only want to focus on peptides. So there's going to be multiply charged species. Anything that's singly charged, we don't care about. So that's lipids, metabolites, drugs, they're all ignored. And the machine is told to just isolate these multiply charged peptide ions for MSMS analysis. And so now that first quadrupole in a second round of analyses will be gating from a list of peptide candidates between 10 to 60 per second for these different peptide masses to go into the collision cell and now experience a higher collision energy, and only the fragments will make it through and be recorded in our flight tubes. So we have our MSMS spectra from each peptide, and that is just rinsed and repeated thousands of times over the run and giving us LCMS traces. So this is a lung amyloid biopsy we had May this year. In the top trace, we've got our chromatogram just showing you that sort of 10-minute run we've got for this particular sample. So if it's a fairly easy sample to work with, we can get away with a 10-minute method. For the more complex ones or the difficult ones, we might employ a longer chromatogram, 20 minutes, 40 minutes, that sort of thing. But at a given retention time, we can see here at 5.07 minutes, we've got a mass spectrum. And if I zoom in on this particular peak here on the x-axis, we can see the spacing of those carbon-13 isotopes. It tells us what the charge state is. So this is a gap of 0.5 between isotopes, which means it's a 2+ ion, and the software has obviously worked it out and told us in brackets, this is a 2+, it's therefore a candidate for MSMS. And so this bottom panel is now all the fragments derived from that species, which has been gated in that first quadrupole for fragmentation. And so this is a hemoglobin peptide. We can see the spacing between these fragment ions corresponds to the different masses of the different amino acids. If we read from the right to the left, it's MFL, SFP, TTK, which apparently comes from hemoglobin alpha. And so you can see in this lung biopsy, it's actually been one of the more abundant signals in the sample. So I dare say all of these prominent peaks actually came from hemoglobin. And our task is to look beyond that and try and find those amyloid signals that will be further down the list, hiding in the smaller peaks in this chromatogram. So yeah, blood is a bit of a problem for us, but sometimes in the biopsies we have to go where the blood vessels are, which is where the Congo red staining was. So we can't often escape from this particular issue. Luckily, we're doing this all with software. So we've got this package called Protein Pilot that lets us do the database search. So we search against a database of all the human protein sequences, and we've added in probably a few hundred IG sequences of various types to try and give us a bit more diversity of immunoglobulin sequences to look at. And here we've got a lymph node amyloid biopsy that we had. There's a list of proteins in this top panel. Number 14 in the list here is IG lambda. And if we click on that, we get this middle panel showing us all the different entries that share some or all of these peptides in this middle sequence column. And we get to see if there is anything that's been cleaved aberrantly. So we're looking in this column for signs that an enzyme other than trypsin has operated on the protein that's been found, because that indicates a long residence time in vivo and being degraded by endogenous proteases, which is a factor that correlates really strongly with amyloid monomers. That's a very key thing we keep our eyes open for. And so here you can see there's a few rows where there's aberrant cleavages stacking up. Additionally, you can allow the software to look through and perform in silico mutations of all these entries. And that's how we often get more matches to the antibody sequences that we don't actually have in our database. We tell the software to do in silico mutation on the fly while it's searching and allow up to 2 amino acids per peptide to be switched for any other 2. So that helps us spot things that aren't actually in our database but exist in the patient. However, the caveat is a lot of mutations can be in actual fact, artifactual modifications like methylation, formylation, methylol. So we have to be cautious about interpreting this evidence proposed here and just have a bit of a grain of salt when we look at those proposed mutations. But it is a useful source of information for us, as we'll see in a slide to come. The bottom panel here is just a sequence coverage map showing us for Ig lambda. In green, the peptides we feel very confident about. In yellow, the ones where the scoring has been a bit more modest. And then in red, the poor scoring matches. And in gray, unobserved regions. Excuse me. And looking a bit more deeply at this Ig lambda match. So if we click on that protein, we see these very highly accurate masses for the precursor peptide measurement. It's all in the sort of 0.00 something mass units, which is very encouraging. We want to see really specific information we can trust. And down here in this bottom panel now, if I click on one of these peptides, it shows you the table of theoretical masses you should see if you fragment this peptide. Whereas in the right panel, excuse me, we have the observed masses that we saw in our MSMS measurement, and we're hoping to see a lot of green matched peptide fragments that are aligning with these theoretical masses. The first two columns are the fragments that contain the peptides N-terminus, while this last column, the Y ions, are the very prominent ones that contain the C-terminus and usually the basic residue that trypsin leaves at the end. Excuse me. So if we just zoom in for a second, one of these to look at the fragment data, this one at 634. If we zoom in to just a Dalton and a half window here now, we can see that in the table, the theoretical mass for this fragment is 634.3406. And here we're looking at an observed mass that's within 0.0004 AMU for our fragments. So even the fragmentation spectra are really highly accurately measured. It gives us really good confidence in our peptide identifications. So it gives us great specificity and selectivity. A kidney biopsy from, we have this match here, number 18 on our list of proteins, fibrinogen alpha. It's a suspicious character. We know that it is possibly an amyloid forming monomer. And so we know that that's normally the case when there's a mutation. And in this data set, and amongst all the peptide matches, there were a few that all pointed towards a very particular known amyloidogenic mutation. So again, this table shows us the theoretical fragment masses you should get for the sequence. And E545V is meaning that this third to last residue of this peptide, glutamic acid, has been converted into a valine. And so now we know that we should see all of the masses above that value have all been shifted by the same amount, 29.97 Daltons. And so, yeah, we can see that these prominent fragment ions are appearing at the expected mass shift if there's been a mutation at that particular site. So it's really specific information. There's lots of evidence all pointing towards the same thing. It's very unequivocal. So that's quite comforting to see. We take our data from that software package and apply an R script, an in-house R script written to help us pull out information about a subset of proteins that we hope to look at more closely in terms of how many peptides we saw, what their summed intensity of all their peptides was, if there are any semi-tryptic cleavages that we obviously are really keen on seeing. And we've allowed a few more metrics to be collated, which we aren't making huge use of just yet, but they've been recorded for posterity. And what we hope to see in here is that there's a combination of really high summed peptide intensity for that protein, along with a suspicious number of semi-tryptic cleavages indicating that it's been degraded by proteases in vivo. So this is a really clear-cut case. And as I say, we actually had, I think it was 7 or 8 peptides maybe that pointed towards that mutation. So really, really confident stuff. But just to sort of contrast that with a cardiac amyloid biopsy, this is one where the clinicians were asking us, is it going to be ATTR or AL? This one, pretty convincing. We've got a lot of signal here for Ig kappa, and again, 7 of these aberrant cleavage events going on. So that's a pretty clear-cut case for that outcome. Whereas a different cardiac biopsy, again, the same question presented. We were looking for evidence of one of those two. And of course, in this case, we had 113 different peptides, all pointing towards transthyretin. 30 of them had been degraded by some other protease. So again, a pretty much unshakable conclusion. And I think this one was for a patient looking to be eligible for that magnitude gene editing trial. So this is, yeah, pretty clear-cut evidence that they were indeed eligible for that one. We had a case a little while ago where we had that list of proteins that we were searching through for evidence of a particular monomer. We were getting nothing. And at that time, we didn't have this last row on our table. But luckily, the Ask script we've developed has got a tab where we ask it to go through all of the proteins, regardless of whether they're known amyloid or not. And for them, it was a real, that particular case, it was a real enlightening moment because we saw top of the list with 24 of these unusual cleavages was this peptide or protein called natriuretic peptides A or ANP, I think in medical parlance. And so that one is on our list of proteins to look for from now on. Haven't seen any since, but that was a real good indication that this backstop that we have built into our script will help us spot things that are unexpected or novel or unusual. So we get a chance to look for things that are not the usual sort of flavor. This is the sort of general sort of final page of the report that the clinicians will probably look at. It's obviously got our main findings where we describe what we've seen and the evidence that we've considered important in terms of calling the monomer. This is that case from the renal biopsy with fibrinogen alpha. We're trying to give a sense of just how abundant this is by showing that, there's 10 of the 30 most abundant peptides coming from this one protein, which is really, exceptionally unusual. And then in this case, yeah, it's important to point out that we've already pinpointed that there is this known mutation in our data set. But of course, they're welcome to go and screen that gene for any other mutations if there are more than one. I don't know. But yeah, that's our main findings. In the second section, we've got all our different other candidates. We've just been open about displaying what other proteins we've been looking for and that have lost out in terms of being called the monomer. And then finally, just our summary to sort of get to the cut to the chase and call the case. This table, kindly put together by our colleague, shows the sort of array of monomers that we've picked up over the years since we started this. You can see it's dominated by the light chains and the ATTR, but there's been a few other unusual ones. There's the ANP one that we just mentioned. And so in graphical form, half of them probably are, light chain, a good chunk at ATTR, and then there's a slew of other little things. There's been about four, I think, of the AFib, and an unusual ApoA4. But then, yeah, we've also got cases where we aren't getting something that we consider definitive enough. Other groups overseas might be quite happy to sort of look at just the intensity of certain proteins that are in the biopsy. But we do feel kind of cautious about calling that without having that additional backup of seeing those aberrant cleavages in the matches. So yeah, maybe we're cautious, but we're also not, for these ones, we're not really in control of how much material is presented to us or how much amyloid is present in that material. If we can identify, 300 or 400 proteins in a biopsy, but not see anything particularly suspicious, then maybe the amount of amyloid in that tissue is below 0.01% of the total mass of protein, and we're going to struggle when that sort of dynamic range is so wide. So yeah, we feel we've done pretty well to get about 85% of the time something quite clear cut that we feel confident about bringing forward in our report. But there's always challenges. We have had cases where we've questioned whether or not the Congo red staining was convincing, and upon reflection, the team have said, yeah, we actually overcalled it. So yeah, we're not in control of those variables, but yeah, we do our best to get a result as often as we can and as convincing a case as we can present. So that's my presentation. I hope it made sense to you all, but open to questions.
Oh, can I ask what's the the false positive rate?
We don't actually really get to have a measure of that. We are sort of
Yeah.
we're left with a case of um, we've called something and it's information only. Yeah, we don't really have a chance
Yeah.
to sort of have a definitive confirmation by some other technique. Um,
Because it's, I mean, from a clinical perspective, as you know, it's really useful to have AL versus TTR. And, there was one case recently that was discussed here who, they often have a positive BPD scan. So you're thinking it's TTR and the mass spectrometry comes back as AL and that'll define treatment. So is it, when you get a report that says this is AL, it's pretty confident
we are naturally
that
quite
that's what it is.
cautious. I think we're
Yeah,
probably more cautious than others. So that might account for some of those ones where we haven't made a call. We've
yeah,
just not had enough
yeah.
evidence to say for sure. I mean, Peter
If
and
you've
Garth,
made a call, you're certain.
well, Peter and Garth review our data and they also get to review the clinical information around that patient. So they'll see the histo notes and maybe other information as well to sort of bolster the case. So yeah, I think between yeah, our data and other information around the case that's available to Peter and Garth, we hope we're not missing anything key or obvious.
Because the result often guides treatment.
Yeah, that's foremost in our
Treatment's
minds. We know
wildly different. Yeah.
yeah, we're obviously aware there's a bit of responsibility with this. And so yeah, we are interested always in improving what we do. There's probably a few things that, can be investigated as ways to improve practices. We're not saying we're the finished product, but yeah, we do feel that we've got a fairly good system to screen these cases. But yeah, I'll let Peter comment if he thinks there's any other directions that we need to go in to somehow validate. We don't actually have any negative control information. There's no tissues we can work on that are sort of matching healthy heart, liver, kidney, lung. So all we've ever received have been the amyloid cases. And as you say, often there is some conflicting information that maybe is ambiguous as to what they feel the monomer might be. And sometimes we do probably have information that seems to go against some of that. So yeah, it's a big call to have to make. So we want to try and be sure when we do make those calls.
It's much appreciated. Our email, my emails with you. Has anyone else got any?
Well, Martin, that was a tour de force. I'm beyond impressed, actually. I mean, I had a very superficial understanding of the process, and I didn't realize it was actually there were 10 times more steps than I understood, let alone a whole raft of acronyms that I didn't understand. So yeah, quite incredible. It does strike me. It seems like $1,100 or whatever it is now seems very cheap.
Well, we try and keep it simple. I mean, I was just looking at a paper before I came on it. There's a German group that have done a bunch of validations of their mass spec technique. And yeah, their sample prep seemed to be like 3 times more involved than what we do. And the analysis times were probably 2 or 3 times what ours are. And I just thought if we had to do that, we'd have to charge like triple just to sort of replicate that
Mm.
complex workflow. And it didn't necessarily seem to be delivering much more. So we try and keep it as simple as possible to minimize, the steps, minimize the handling. Yeah, keep it as simple as you
Mm.
can as our mantra.
Yeah, I noticed it was very simple.
Mm.
Yeah.
And we aren't using laser capture microdissection. The obvious question is, could you do it better with that? But we don't have easy access to that. It may exist up at Grafton. I'm not quite sure what it would cost us to access that and how much time delay that would introduce between, us coordinating tissue going back and forth. So yeah, it is a simplistic way of, harvesting tissue, ideally guided by the clinicians pointing out which regions we should focus on, which really helps us. Um, but yeah, it is kept as simple as we can and as affordable as we can. Yeah.
How much do you rely on seeing the amyloid sort of matrix constituents, if that's the right word, as part of the picture?
Yeah, some people do obviously propose methods, mass spec methods, where they do rely on ratios of
Mm.
intensities of, say, lambda versus serum amyloid P component. We haven't been creating those metrics. It's encouraging to see them. It makes you feel like, yep, we've got actual amyloid solubilized from the tissue. Um, But no, we do tend to focus much more on what features are just, aberrantly high in abundance and have got those signature diagnostic non-tryptic patterns. That's sort of the main thing we kind of hunt for.
Yeah, when I was on my sabbatical, and which is 6 years ago now, at the NAC in London, then they were quite keen on picking out those matrix constituents. And so it did sort of feel, I didn't go into it because I didn't understand it well enough to go into it, but it did feel like they had a slightly different approach. But they were actually quite new in their sort of implementation of that.
Yeah, many do just rely on intensities. I think, some publications do seem to rely on, I wouldn't say simplistic, but yeah, fairly,
Basic.
yeah, they do seem to rely on intensity rather than anything else, which seems to me slightly open to other conditions that are happening at the same time in that tissue, like in terms of inflammation or antibodies, simply giving you that signal or MGUS, where you have got suddenly a lot of antibody showing up, but it's not really part of the deposit, it's just there. So I do have concerns about those simpler tools. Obviously, ApoA4 is often put forward as a matrix component, but it can very occasionally end up being
And
something more
amyloid.
important,
Yeah.
as we've seen recently with one of those cases being an unusual one with that signature of degradation features.
Yeah. Well,
Thank
doing a
you.
great job. Thank you.
Yeah, yeah, it's much appreciated. I
No
think we've got
problem.
a couple of cases